Hi, Suzuki Science. So, grade 12s, here's that promised lesson on Sanger sequencing. I will not test you on it as I mentioned before, but it is a genetic technology, and in the final project you might be able to incorporate some of the learning from this particular technique. So, when we talk about biotech, we're doing a whole week, a section on biotechnology, and we're not there yet. So, when we do talk about biotechnology, recognize that this concept falls into biotech. At one point, we were trying to determine what was the words, the dictionary of the human genome. What does our DNA look like from beginning to end? If you can label chromosome number 1 as the beginning and chromosome 20, the sex chromosomes, as the end. And we thought that if we're able to read the book, at least identify all the letters in the book that makes up our DNA, then we would have a better chance of understanding, things that make up our characteristics, our genotypes that display into our phenotypes, diseases and corrections and all that kind of stuff. It turns out that even though we do know all the letters, it's kind of like looking at an ancient script. We might be able to recognize all the characters that make up this ancient script, but we're not able to decipher what those letters mean together. And with the DNA sequence, they did sequence it, and on the screen here you can see an abstract from one of the original articles of when they sequenced the human genome, and this was written in February 2001. So we have 3 billion, and even though that number in the grand scheme of big numbers is not that big, it's a huge number. And we, as humans, were able to spend about, I think, $3 billion. Wait, what was it? I don't remember what the actual cost was, but it was pretty expensive. I think it came down at $2.7 billion. And in that time, they're able to determine the actual sequence of letters that made up our genetic code. They sequenced the entire human genome. This took a long time. And now we're able to do something very similar in much less time. So the technique that was used to sequence the human genome here is different than what I'm going to talk about. There is an organization called the National Human Genome Research Institute, the NHGRI, and they do a very good job of explaining the Human Genome Project. So I'm going to leave a link on the resource document so you can come here and just kind of skim through it to get a better feel for the big picture. I also have a video sequence from TEDx. It's a TEDx lesson that you can work through at your own leisure if you'd like to learn more about the Human Genome Project. I'm going to focus on the skill of sequencing. So, sequencing involves determining every single letter in the genome of an organism. A human's been done, but there's still many other organisms to be done, and we've only done several humans. We haven't done them all, and each person's genome looks slightly different. You might have heard of things like Ancestry 123 or DNA and Me. They sequence your DNA and then they compare your DNA to these big giant databases to determine where your ancestry came from and what diseases you might potentially code for. We talked about some of these things in grade 10. So on the screen here I have a nucleic acid. It's DNA. You can tell it's DNA because it's got the phosphate bound here, and there's no OH here. So DA stands for deoxyribo. Deoxy. Ribo or ribose has an oxygen hydroxyl group here and a hydroxyl group here. Carbon 1, 2, and 3s. Deoxyribo is missing an hydroxyl group on carbon number 2, but it has it on carbon number 3. And if you recall from the beginning of replication and from way back in biochemistry, that hydroxyl group on carbon number 3 is incredibly important. We need this. This has to be here. Why? Because that allows an incoming nucleotide to bond. The carbon number 5 will bond to carbon number 3 and the chain will grow. That's the process. Okay. Now, if that hydroxyl group was missing, right? If it was missing, if that didn't exist, so if I were able to erase it, so I'll just cover it with so let's say there's no hydroxyl group here. If there's no hydroxyl group here, what that means is that when this comes in, this phosphorus tries to come and bond, because there's no hydroxyl group, there is no possibility of that condensation reaction. Without that condensation reaction, this cannot bond. So this leads to a very interesting possibility. As long as you have a normal hydroxyl group, these things will bond. Everything's perfectly fine. But if I were to grab this thing over here and rotate it around and bring it in, again, it would be perfectly fine. But let's say that we take off this hydroxyl group. So now what we have here is called a dideoxy. It's missing 2 oxygens. So it's dideoxy. So here I have a dideoxy A, TP, a dideoxy T, TP, a dideoxy GTP, and a dideoxy, well, this one's got it still on, we just got one, this on here, a dideoxy CTP. Collectively, they're known as dideoxy NTPs. Deoxy NTP would be these, N being the nitrogen base, and DD being dideoxy. So if one of these were to bond, then the entire process would terminate. So this would come on, it would bond, and then nothing else could bond. It would stop right there. So this leads to a very interesting possibility. So remember that as we move forward. In order to do the sequencing, it's a replication process. You need all the stuff you'd use for replication. So you tell me, what do you need to replicate? And then I'm standing at the front of the class and you'd be responding, or if we're doing conferencing, you guys would be typing the answer. And one of the things you'd probably tell me is, oh, if you want to replicate, then you have to have a DNA template. You must have a DNA template in order to replicate it. So let's go with blue here. You need a DNA template. All right. And what else do we need? We also need a primer. Right? We need a primer. So we need a template. And we need a primer. Okay. And what else do we need? We also need nitrogen bases, right? We need A's, T's, C, and G, right? These are collectively known as DNTPs, nitrogen triphosphates. Okay. What else do we need? Well, we also need a polymerase, right? We need a polymerase. So, which polymerase is it? DNA polymerase or RNA polymerase? It's going to be DNA polymerase. Right? I just got a new pad, so I can actually write. Okay. So these are the ingredients for replication, along with a bunch of other things, right? But these are the main ingredients that we need. So as long as you get the DNA polymerase, you got a primer that's already created. The primer is already being constructed to attach to the template properly. You have to throw in a whole bunch of nucleotides, and we're good. But you need one other thing. We also need our dideoxy nucleotides, right? So I'll do this in green. So we need the special A, we need a special T, special C, and special G. And these are going to be our dideoxy NTPs. We call them DDNTP. So these are the ones, so this one over here, they have the oxygen group on it, right? They got the oxygen, they got the OH, got the OH, the OH, and the OH. These ones over here, they don't have either the carbon number 2 all-hydroxyl or the carbon number 3 hydroxyl. So these are dideoxy NTPs. So here is the ingredients that are necessary for Sanger sequencing. Okay. Now, once we have the ingredients for Sanger sequencing, then we're ready to kind of run the experiment. So the experiment involves really 4 test tubes. Okay, so there's 4 reaction vessels. So test tube 1, we'll call A. Test tube 2 is the C reaction. Doesn't have to be this order. Test tube 3 will be the T reaction. Test tube 4 will be the G reaction. So I got the A reaction, C reaction, T reaction, G reaction. It's a weird order. Okay. And so in here, you're going to put everything. You're going to put the primer. Let me grab yellow. So we need a primer in here with its template, right, in here. The DNTPs go in here. The DNA polymerase goes in here. And in this reaction vessel, I'm going to put some dideoxyases as well. So it's all going to go in here. In this test tube, it's going to be all the same things, except this time, instead of A, I'm going to put in C. So C. So there'll be some of the C in here. So C will come in along with everything that you see here. T will have the dideoxy T with everything, and G will have the dideoxy G with everything. Okay, so let's focus on this test tube right here, the A reaction. So we've got all the ingredients, we've got the A reaction, so now we're ready to go into the experiment here. So let's go back to black. Black. All right, so we're going to call this the A reaction. So in the A reaction, the primer is set. So I already have the primer. The primer is a complementary sequence, A, G, G, C. There's no, if I had the primer and there's going to be a T equivalent, it would be U. But there's the primer. And so now this is going to undergo this replication process. And so here's my polymerase. I'll draw a polymerase in here. Okay. And I'll use blue to represent the things coming in. So this polymerase, it's going to bond thanks to the primer, and it's going to move along. And as it moves along, it's going to transcribe this DNA strand. Well, it's going to replicate, sorry, this DNA strand. So wherever you see a T, you're going to get an A. Right? But sometimes so you're going to get an A. But sometimes it's not going to be the deoxy A that's going to bond. Sometimes it's going to be the dideoxy A that bonds, right? And if the dideoxy A bonds, then the whole thing stops. The whole thing will stop. So let's do the next reaction. So in the next reaction, let's say a normal A does bond in the next reaction. So normal A bonds. And into this A, you're going to get a T. And in this C, you're going to get a G. And this A, you're going to get a T. And this T, you're going to get an A. But let's say this time, instead of a normal A, sometimes you're going to get this dideoxy A. When the dideoxy A bonds, it's going to terminate. Right? So let's do this all the way to the end, all the way to the logical conclusion. I'll do one more in front of you. Grab one more. Right. And as I'm doing this, you know, to say it out loud with me. I can't hear you, but you can hear yourself. So I'm going to get an A, a normal A sometimes. T, G, T. Sometimes a normal A will bond. To that G, I'll get a C. To that A, I'll get a T. And to that T, I'll get N. Sometimes a dideoxy A. So we'll stop. Right, so now let's count these up. How big is this one here? This is 1, 2, 3, 4, 5. 5 big. This one here is 6, 7, 8, 9. This one here is 10, 11, 12. This one here is 13, 14, 15, 16, 17, 18. This one is 19, 20, 21, 22. 23, 24, 25. 26. Right? So I end up with a bunch of fragments. One that's 5, one that's 9, one that's 12, one that's 18, one that's 22, one that's 25, and one that's 26. I just didn't copy this whole blue thing down. So what does that mean? Well, wouldn't it be brilliant if we had some kind of technique that allowed us to separate the DNA fragments based on length? Okay, let's remember that. So as I keep on going through this, I'm also going to do a T reaction. I'm going to do a C reaction. I'm going to do a G reaction. Now, if we had some method that allowed us to separate these based on length, then we would be able to determine the order. And as a matter of fact, we do have that ability. Imagine we had the ability to read this. Well, this special technique, which is something we're going to learn about during our biotech lessons, is this thing here is known as a gel electrophoresis. And what a gel electrophoresis allows us to do is it allows us to separate DNA and proteins, if you want, amino acids and proteins. It allows us to separate it based on, drumroll, length. The shortest ones are up here, so these are the shortest, and these are the longest. So the shortest one is going to be a T, and then the second shortest one is going to be an A, and the next longest one is going to be an A, and C, and then G. So if I read this through, I know I'll have my primer, right? I'll write down the primer, whatever the primer happens to be. I don't remember what the primer is, so I guess I'll go back and take a look. AGGC. AGGC. And we know there's a T. I just didn't have it there. But now, oops, not T. A, sorry. I know there's an A. That's probably supposed to be part of the primer. Okay. And now I'm going to read through. So next, the shortest is going to be T, and I read the next two, A, and I read the next one, C, G, there's my sequence. I read it off, but what you need to recognize is that this is actually the sequence that was the complementary sequence, right? Because these are all complement. This is the original sequence here, and we have these complementary sequences. So the thing that's on this gel here is actually the complementary sequence. So now I have to take the reverse, right? The complement of it. And so I just write that out. C, A, T, A, T, C, G, G, A, T, G, C, A, T, C, G, T, T, A, and then T, G, C, C, T. So I probably wrote their primer backwards, to be honest. But this part, it's not important, right? So I'm just going to scribble this out. Because the primer, we already know. What we are learning is this sequence. So this here is the original sequence. And that is how we sequence, right? So I showed you specifically how we do the A reaction. And so remember, the deoxy will bond most of the time, but occasionally you'll get a dideoxy. If a dideoxy bonds, then it stops the whole process. And since this A occurs in several different spots, you end up with all these different lengths. And then we're going to do this for the A, we're going to do a separate one for the T test tube, another one for the C test tube, another one for the G test tube. You're going to end up with basically numbers that go from 5 all the way to 27. And then when you put them onto this special gel, they're all going to separate based on length. And then this can be read, and we can use that to determine the gel sequence, the actual genome sequence of whatever organism we're looking at. Okay, so on the screen I should have put a instruction set that goes step by step what's going to happen. I'll put a link to a video, and you just work through those. The actual worksheet itself is going to look like this. So you're going to see the A reaction, the T reaction, the C reaction, the G reaction. So I've started them off for you already. I've actually put the answers for them. So with this original sequence, I've actually put down the answer so you can double check to make sure you've done it properly. And then once you've worked through this exercise and you've gone through the entire process yourself, and you put it on the gel, then you can try it on your own with this. new sequence and try the same thing. Right? So it's just practice. It's just a technique. You won't be tested on it. You might make reference to it on the final project, but you won't be tested on it formally. Okay? That is Sanger sequencing. Thank you for listening.